Hello, everybody. Unfortunately, I have to give this presentation as a recording because for me, the Nanion user meetings are somewhat cursed. 2 years ago, I got sick from Corona. Last year, I got a pneumonia, and this year, I have teaching. So I do apologize, and I really wanted to be here. Nevertheless, without much further ado, I'm going to tell you a little bit about substrate recognition and transport mechanism of the pin-formed auxin exporters. So auxin is a hormone in plants, and when we say auxin, we actually mean a group of compounds that all have growth promoting effects. And generally, this is synonymously used here with indole 3 acetic acid, which in most plants is the dominant auxin. Polar auxin transport controls many processes in plants and plant development. And this is shown here. So embryogenesis in plants is due to polar auxin transport, also lateral root growth, etc., etc., and a couple of other very important things. This, in turn, has led to huge efforts on parts of the military, mainly in the 1940s, to look for auxin herbicides, which are shown here. And to date, auxin herbicides still make up to 20% of global herbicide application. And some of them are really heavily and widely used, such as picloram and dicamba. But they also became notorious because the phenoxycarboxylic acids, including 2,4-D and a couple of others, became used during the Vietnam World War in formulations such as Agent Orange. Auxin is moved in plants by a number of transporters, and this is shown here in this picture. And auxin is moved into the cell across endomembranes and so on and so forth. But the most important part for polar auxin transport is this group of transporters down here, the so-called pin-formed auxin exporters, which I'm going to talk about. And they are important because they are underlying polar auxin transport, because in the cell they are polarly localized. And what you can see here is a collection of PIN mutant phenotypes. This here is the PIN1 mutation that gives rise to this pin-shaped inflorescence. Closely related genes such as PIN2, this is shown here, these seedlings cannot grow towards the gravitropic vector anymore, and a PIN347 triple mutant fails to grow towards the light. So these are typical auxin responses, and pins are underlying all of these mutations. A couple of years ago, Björn Pedersen in Aarhus and I joined forces. Björn Pedersen is a structural biology lab, and he became interested in the pin proteins because nothing much was known. Still to date, very little is known about plant transporters in general, and particularly hormone transporters. And they used PIN8 mainly because they could express it to interesting levels. They could purify it to homogeneity. And as you will see later, they could also solve the structure by single particle cryo-electron microscopy. The part of my group was to establish the activity, and this is also why I am speaking here now, because we did that mainly using solid-supported membrane-based electrophysiology using the Nanion surfer, and this has proven to be super useful for us. So the first thing that we did was we checked the IAA dose response, and what you see here is the typical on peak that you can get in a surfer signal, and you may appreciate or may have heard in talks other than mine in this section that generally what you look at is the peak current, because the peak current is as close as it gets to a steady state signal, because on surfer you don't have a persistent steady state signal. And what you see here is that we tested several concentrations of auxin, and this here is a concentration dependent response. And when you plot the peak currents here, what you can see here is you can fit a Michaelis-Menten kinetics, and this here, the triangles is on empty sensors, so nothing much happens. So we got a Km, or better, an EC50 of around 350 micromolar. This was super surprising to us because before we had done characterizations of PIN proteins in Xenopus levis oocytes, and this here is also PIN8 in Xenopus levis oocytes. And as you can see, we got here a linear relationship between the substrate concentration and the export rate. But what you may appreciate is that we only tested here up to 10 micromolar. In planta, 10 micromolar IAA is a sublethal dose. So we were shocked to see that our transporter only has substrate affinities in the lower micromolecular range, which means that in the physiological range, and what we had looked at so far, is here in the very, very, very left part of the curve. For a long time, I thought there was something off about our data, but a couple of months after we published this story 2 years ago, 2 Chinese labs independently published other structures from member of the PIN family, and they also found these very, 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 very high substrate affinities, indicating that PIN proteins work, way past the physiologically relevant concentration. And in vivo, we are looking at a linear relationship between the substrate concentration and the transport rate. So nevertheless, what we also did is, using Surfer, we characterized a couple of compounds that were known in auxin biology to play an important role. And as you can see, we got current responses for some. We didn't get current responses that we would consider transport or that being a good physiological substrate for the PIN proteins. And this was all very nice because Björn now knew that we had functional protein and that did something that was known from the literature. And so they went on and could determine the structure of the PIN proteins by single particle cryo-electron microscopy. They got 3 structures: outward open APO without the substrate bound, IAA bound outward facing, and inward facing with a well-described and long-known inhibitor of auxin transport. And what you can see here is, this here is the inward open configuration. This is the outward open structure. So, and you can see that there is no passage through the protein in the inward open configuration, and there is no passage here through the protein in the outward open configuration. What you can also see and may appreciate, so we have a dimer, and one monomer is shown in white and the other one is shown in blue and green. And we named the blue part here the scaffold domain, and this here is the transport domain. And the 2 monomers interact here at the scaffold domain. I will show you a movie about the transport mechanism at the end of the talk. But suffice to say here that this fold is the same fold as the proton-sodium exchanger that you will also hear about here in the same session. So the fold and mechanism of PIN transporters is shared by a number of other transporters. Using the structural data that Björn's group determined, we then did point mutants, and we looked at the effect those mutants have on auxin transport mediated by PIN proteins. And what you can see here now is the IAA molecule in the binding pocket. And what we have here on the other side is a pseudo-symmetrical support site. In the sodium-proton exchanger, this is the part where the sodium binds. In our case, this is not sodium, but it is a structural water that is in there. And as this here is also highlighted, you can see here, so this here is the pseudo-symmetrical support site is part of the transport domain, whereas here a large majority of the binding of the substrate is then also due to the scaffold domain. And when we made point mutants, turning them either to alanine or we did isosteric mutations, we could determine which residues are important for substrate binding. And it turns out that when you touch here the pseudosymmetrical support site that influences the transport mechanism, you kill the protein dead. However, when you look at the residues that are outlining the substrate binding pocket, what you find is that this is pretty much forgiving. I mean, you can reduce affinity. Some mutations even don't have an effect. But nevertheless, I mean, mutations in this section are quite forgiving. And when you think about it, that actually makes sense from an evolutionary standpoint, because the transport mechanism is something you really want to conserve. But if you want to be able to adapt to substrates, and evolution can drive other substrates here to be accepted by this transporter, then it actually makes a lot of sense that these mutations are somewhat forgiving. So obviously, then we became interested in asking the question, what does it take to be a good substrate? And what we have here now is the binding affinities. And then these are ranked. You have IAA here on this side. And then we have a couple of substrates that we tested. And what you see here, what I highlighted, are well-known phenoxyacetic acid substrates. That are shown here in dark gray. And then we did a couple of things. I mean, we changed the position of the chlorine residues. We substituted this chlorine residues for methyl groups. We got rid here of this ether in the side chain and so on and so forth in order to understand what does it take to be a good substrate. And it turns out, in a nutshell, that what you need is a carboxylate. You don't need this oxygen here. And then it's really, really good. And the more chlorine residues you have here in this ring structure, the better the substrate is, because obviously somehow there are hydrophobic interactions here with this p electron system in the substrate binding pocket. But I want to share with you a little bit what we did here, because it's actually interesting in terms also of surfer. And we looked at a couple of these substrates, and one of them is 4-CPA, and this is a super lousy pin substrate. I mean, here the KD is super, super low. I mean, KD is also lower than KM in our case, but nevertheless, this is really, really a very, very low substrate affinity. And we focused on 4-CPA for a while because we and others had shown that it has a very high potential in transformation of recalcitrant woody plants. So nevertheless, we looked at this 4-CPA and we found, well, it's a pin substrate. Then we also looked at the most notorious and prominent of these auxins, which is 2,4-D, that has 2 chlorine residues in the ring system. And we found that also 2,4-D is a pin substrate. And what you can see here, this here is the on peak of IAA, and this here is the on peak in green for 2,4-D. You can see that there is a little bit of difference that is mainly concerning the width of the peak. However, when we titrate this, what you can see is that these dose responses are virtually indistinguishable. What you always see here in the back is to show that also the 2,4-D current is sensitive to a well-known inhibitor, but this doesn't matter for the purpose of this talk. So the challenge that we were facing now is this binding or is this transport? And this question is far from trivial, because the surfer signal is hard to interpret, to say the very least. And what you get is a mixture of binding and transport current. And disentangling this is very, very tricky. And I mean, at the Nanion user meeting, Andre Bazzone will be around. If you're really interested in doing this, grab him. It is a very, very hard thing to do. Nevertheless, one of the things that you can do is, if we have now here the on peak, what we have is 2 components. We have the width of the peak, and then we have the current response here. And this here is the magnitude of the peak current. Now, this here is at an LPR of 10. When we vary the protein concentration and we change the lipid to protein ratio, you can do a couple of different things. So what you would expect is that if you have transport, what changes is the width of the peak, because the amounts of molecules and substrate that can go into the liposome until you reach equilibrium is going to be dependent on the amount of protein in the membrane. However, when you only have binding, this peak width will not change. The magnitude of the peak current will change, but the width of the peak will not change. And we had to do that, and we had to suffer from that, because usually what we do in order to disentangle binding from transport, we use a different line of evidence, and we use Xenopus levis oocytes, and we look for radioactive transport of substrates. However, with all these compounds, we had to look at so many different compounds, and they aren't available commercially, and to synthesize them is super expensive. So we tried our best. And doing our best here also to look at the time constants here of the peak width requires purchasing the 1.5 square millimeter sensors from Nanion to get a better resolution in the time range. And what we get here now is, this here is an example for 4-CPA. What you can see here is this here, is at in gray, this here is at an LPR of 10. And what you can then appreciate when we here use the LPR, and now we used relative currents to get them on the same side, and also here to show that what changes here is the full width half maximum constant. So you can still see that here is a difference. So we get an increase in the time constant when we reduce the amount of protein in the membrane. What you can see is we did that here for all the substrates. We did that for IAA, we did it for 2,4-D, we did it for 4-CPA, we also did it for 2-CPA, another of these compounds. And what we always get is that we get an increase of the full width half maximum, so the time constant of the peak, which is consistent with this being transport. The important part here is this is NPA, which is the inhibitor that we had used in order to get the inward open structure that has been used in auxin biology for decades. And what you can see here, we do not get an increase. So in this case, the time constant remains pretty much solid. So this indicates that what we get here for NPA is a binding current, and in this case it's a transport current. So we could show that not only do these herbicides bind, they're also transported by PIN proteins. All right, we then proceeded, and Björn determined the structure of pins bound to 4-CPA and 2,4-D, and this is now for structural biologists something that is very, very interesting, because we got many different structures. And what you may appreciate is we have here a pre-binding state where the substrate is still not flipped into the binding pocket, and we got then here another structure where it is in the inward position and in the outward position in different positions of the structure. That was very informative, but the most interesting part for us was that you can see here that one monomer is outward and another one is inward. Then we got here outward configurations, and here other types of configurations, which means that in our case, we could show that the monomers in the PIN dimer can move independently. This is pretty interesting because this was not known for any of the proteins that have the same fold as PIN proteins. Now, the other thing is, and this here is also the power of Surfer again, we again did point mutations that then were identified to be critical for substrate coordination here in a pre-binding state, and then for substrate binding in a binding state. And we determined the KD values, we determined the EC50s, and so on and so forth. And what you see here now is a heat map, and this is all normalized here to the wild type protein. We then are able now to obtain mutants that transport the normal endogenous auxin as expected, but have much higher affinity for the herbicides. And on the other scale of the spectrum, we now also have mutants now that are virtually blind to the substrate. And this is super interesting because these types of mutants are then interesting for the chemical industry in order to design herbicides that have better effects on plant biology, and perhaps we can even find herbicide resistant plants, which would be really, really helpful in our case. So the last thing that I want to show is, and this here is now running, this here is again one monomer in white, the other one is here shown on the left side in blue and green, and when it loops again, what you can see is the auxin molecule binds to the inward open position. The carboxylate is coordinated here by an asparagine residue, and the interesting part then is that the substrate has to flip in the binding pocket, and once it has flipped, the protein moves. It's a rigid body movement, so the transport domain moves by 5 angstroms, and then we have the release of the substrate on the outward side, and we get the flip back. And when it loops again here, these residues, this here is the pseudo-symmetrical binding site of, and this movement back is triggered here by the pseudo-symmetrical support site. All right, with that, I should acknowledge the people here very, very briefly. This is mainly the work of Lukas Schulz. He was supported by Sarah Kutnik and Dorina Janacek. And the structural work is done in Björn Pedersen's lab in Aarhus, and it is mainly carried by a very, very talented postdoc, Kien Lam Ung. And with that, I do apologize for not being able to join you in person. If you have any questions, I try to be around on Wednesday, and if not, please drop me an email. I'd be happy to chat with you and explain in more detail.